Okay, hello everyone. I think this is the first time that I'm presenting one of the theoretical lectures in English for our international students. So far, you had an opportunity to see a couple of video materials for experimental part of our biochem course. And of course, you have your practical manuals in biochemistry available throughout the mobile application. So I'm not worried about your practical part of this course. But from this point on, I think that we're gonna publish a couple of theoretical lecture in order to help you with e-learning or self-learning of our biochem course, and to help you, if it's possible, to take and pass the exam successfully when this situation is over. Before I begin with today's topic, I would like, of course, to apologize for my English because I'm not a native speaker, so please forgive me if I make a couple of mistakes while presenting this topic. So I have decided to start from the beginning with the metabolism of carbohydrates, and today's topic is going to be digestion and absorption of carbohydrates. So let's start. Actually, there is a large number of different types of carbs which enter the body throughout diet. And we call all those carbohydrates dietary carbohydrates. But the point is that we should at least remind ourselves about the major types of carbohydrates which are found in our regular ordinary diet. So let's start from the most complex carbohydrates, and let's remind ourselves about the types and the structure of those molecules. So can you tell me which polysaccharides do you know? Which are the most famous? Exactly. So first we have starch, which is a homopolysaccharide containing a huge number of glucose molecules. It is a molecule containing two domains, two parts, one linear part amylose and one branched part amylopectin. Which type of bonds are found in this molecule? Do you remember our general biochem course? Exactly. So we have two types of bonding. First, we have alpha-1,4 glycosidic bond, which is found in those linear parts of starch molecule between two molecules of glucoses. And at branching points, there is alpha-1,6 glycosidic bond present in molecules of amylopectin. Which other polysaccharides do you know? Which is the molecule analog to starch which is found in plants? In animals, it's glycogen. So a very similar structure to starch, but the glycogen is considered to be slightly more branched molecule. Again, it's a polymer of a huge number of glucoses, and again, there are two types of glycosidic bonds found, alpha-1,4 glycosidic bonds and alpha-1,6 glycosidic bonds at branching points. What about the rest of the polysaccharide molecules which are commonly found in our diet? Exactly, it's cellulose, a polymer of a huge number of glucoses connected by beta-1,4 glycosidic bonds. So these are slightly different from the previous bonds mentioned. Apart from polysaccharides, apart from those complex sugars, also a number of disaccharides are found commonly in our diet. Do you know any of those? It's maltose, consisting of two molecules of glucose with alpha-1,4 glycosidic bond found between two molecules of glucoses. Which other disaccharide do you know? Exactly, it's lactose, so consisting of one molecule of glucose and one molecule of galactose. And which type of bond is found? Beta-1,4 glycosidic bond. One disaccharide which is really, I think, the most common sugar taken throughout diet, exactly, it's saccharose or sucrose. containing glucose and fructose molecule connected by alpha-1 beta-2 glycosidic bond or trachalose bond. And of course, there are a number of monosaccharides found in our food like glucose, galactose, mannose, fructose, etc., etc. So now that we remind ourselves about those basic carbohydrate molecules in our diet, let's make a point. So human cells can use only monosaccharides for their metabolic needs and purposes, which means that all more complex carbs but monosaccharides have to be degraded or hydrolyzed to their building bricks. I mean to monosaccharides, glucose, galactose, fructose, and so on. This is completed by the pathway which is marked as digestion of carbohydrates, and it takes place in our gastrointestinal tract. And actually, it's based on the activity of a number of specific enzymes which are responsible to break down or hydrolyze those previously mentioned glycosidic bonds. Further degradation of glucose in cells yields the production of energy, and approximately 50% of total energy needs of our cells are supplied by the degradation of sugars. So let's start from the beginning. So when we eat something sweet or carb-rich food, carb-rich diet, carb-rich meal, the content is in our mouth. So what is going on? Of course, first mechanical chewing or grinding of the food, and it's gonna be the starting point for carbohydrate digestion. Not only the mechanical grinding of food and mixing with saliva, but at this point, chemical degradation of starch and glycogen starts. So there is an enzyme which is responsible for degradation of starch and glycogen in mouth, and this enzyme is called alpha amylase. Alpha amylase is an enzyme which exists in two isoenzyme forms. The first one is called salivary alpha amylase, which is synthesized in salivary glands, and it's secreted to saliva where it exhibits its activity. The other isoenzyme form is pancreatic alpha amylase, which is synthesized by pancreas, and it's active in duodenal juice. This enzyme catalyzes the hydrolysis of alpha-1,4 glycosidic bonds in starch and in glycogen. And like you know from the general biochemistry course, each enzyme requires certain optimal conditions to be met in order to have an optimum activity. So those optimal conditions for alpha amylase are temperature. Of course, the optimum temperature is body temperature 36-37 degrees. Then most of the enzymes require specific pH for their activity. In this case, for alpha amylase, it's 6.7 on a pH scale. And the thing that a lot of students somehow forget to mention is that most of the enzymes require certain activators to be present, and in this case, alpha amylase requires the presence of chloride ions, which are unique activators of this enzyme. When all these requirements are met, our alpha amylase can exhibit optimum activity. But be careful. This hydrolysis is a multi-step process, so it occurs at several steps and several intermediates are formed. Those intermediate products are called dextrins, and here is the list of subsequent dextrins which are formed, each of them with a smaller or lower molecular mass. So at the beginning, when starch or glycogen starts degrading, amylodextrins are formed with a lower molecular mass, and as the degradation proceeds, erythrodextrins are formed, leukodextrins are formed, and acrodextrins are formed. I hope that you remember these dextrins from our practical part of the course when we tested the activity of salivary alpha amylase. So I'm not gonna go into details this time regarding these dextrins and their properties. But let's summarize what happens at the end. So the final products of the alpha amylase activity are maltose and limited dextrins. Usually students remember maltose without any kind of problems, but somehow they skip to mention limit dextrins, and this is why I placed this note, to remind ourselves what are limit dextrins. So they are actually oligosaccharides. If you remember the facts from the previous slide, we mentioned that there is one other type of glycosidic bond found in both starch and glycogen, and that is alpha 1.6 glycosidic bond. This bond cannot be degraded by alpha amylase because it's specific for alpha 1,4 bonds. So the part of the molecule containing alpha 1,6 bond as a central part of the molecule, and couple of glucoses, approximately 6 to 8 glucoses surrounding this alpha 1,6 bond are actually forming limit dextrins. This is the molecule, the one alpha amylase cannot hydrolyze. So we will have to employ one other enzymes for the hydrolysis of this molecule. So in the end, to conclude, the final products of alpha amylase activity are maltose and limit dextrins. So the content from the mouth goes down and enters the stomach. What we know about the stomach and the stomach juice, we know that the pH of the stomach juice is rather low, from 1.5 to 2.5, and due to this low pH, irreversible inactivation of salivary alpha amylase occur. So at this point, at the level of stomach, we have some kind of temporarily pause in the digestion of carbohydrates. So it's gonna continue and it's gonna be completed and finalized in duodenum by the action of a number of other enzymes, and of course in the scope of starch and glycogen it's gonna be pancreatic alpha amylase. So let's move on to duodenum. So what happens in duodenum? First, the content from stomach, gastric juice has to be neutralized. So the low pH has to be neutralized and brought up to pH 1, which is achieved by the secretion of bicarbonate ions from pancreas to duodenal juice. In such conditions, so slightly basic conditions, a huge number of enzymes are active. Not only the ones which are dedicated to carbs digestion, but also pancreatic enzymes dedicated to lipid digestion and protein digestion as well. But we are going to focus on carbs digestion this time. So first I'm going to mention, of course, pancreatic alpha amylase, which is going to complete the digestion which was not completed at the level of the mouth. And we know which are the final products, which are maltose and limited dextrins. So which enzyme is now responsible to degrade or to digest maltose? If you remember, the enzymes which are responsible for hydrolytic processes are called hydrolases, and their names are formed in a very simple way. You just add the name of the substrate and then add the suffix ase. So if we have a molecule of maltose, the enzyme is going to be maltase, or alpha glucosidase, because it degrades alpha 1,4 glycosidic bond of maltose. So it degrades maltose to 2 molecules of glucose, to 2 molecules of monosaccharide. The next final product after the activity of alpha amylase were limited dextrins, if you remember, and one enzyme which name is oligo-1,6 glucosidase is responsible for degradation of limit dextrins to glucose molecules. This enzyme is capable of hydrolyzing or degrading 1-alpha-1,6 glycosidic bonds from those branching points in the central part of limit dextrine's molecules. Then we mentioned other disaccharides. We mentioned lactose or dairy sugar. So if lactose, then the enzyme is going to be lactase, and its chemical name is beta-galactosidase because there is a beta-1,4 bond between galactose and glucose. So we're going to get galactose, the mixture of galactose and glucose after the activity of this enzyme. Then we mentioned sucrose, so the enzyme is going to be saccharase or beta-fructosidase following the same pattern, and this enzyme is going to hydrolyze saccharose to glucose and fructose. Apart from these, there is a list of a large number of other disaccaridases, oligosaccharidases, etc., etc., which are responsible for degradation, digestion, conversion of disaccharides, trisaccharides to monosaccharides unit. At this point, I have to mention the molecule of cellulose with those beta-1,4 glycosidic bonds between molecules of glucose. Cellulose cannot be digested by human gastrointestinal tract because we don't have specific enzymes capable for hydrolysis of the mentioned bond. So anyway, by the enter of upper ileum and small intestine, all complex sugars are converted to their building bricks, are converted to simple monosaccharides: glucose, galactose, fructose, mannose, pentoses. So now those simple sugars can be absorbed from intestinal lumen to enterocytes. And of course, we have to see which mechanisms are involved in such process. And there are some differences between those monosaccharide units. Glucose and galactose are absorbed by active transport. And you know from some of the previous units that this kind of transport requires specific transport proteins, plus it's an energy requiring process. And the mechanism of this transport is concurrent uptake of sodium ions by the terms of symport transport. Other sugars like fructose and mannose and pentoses, they are absorbed by facilitated diffusion. Apart from the absorption mechanism, absorption rate is also different. So, for example, galactose is absorbed the quickest, so the absorption rate of galactose is the highest, and then comes glucose and fructose, mannose, and the slowest absorption is for pentoses. After the absorption to enterocytes, our monosaccharide units has to leave cells, so they have to be transported from enterocytes into portal circulation, and that is achieved by facilitated diffusion. So please, at this point, I'm pointing out and I'm emphasizing the fact to be careful when reading the test question or listening to what your professor is asking you. So you have to focus on whether you're asked about the absorption to enterocytes or exit from enterocytes to portal circulation. So by portal circulation, our monosaccharide units are brought to liver, and you know that liver is some kind of general distributive center of the whole body. So liver is going to decide what is going to happen with those sugars. Are they going to be absorbed in the liver and incorporated in the molecules of glycogen, or they're going to be distributed throughout main circulation to target cells and tissues where they're going to be used for other purposes like obtaining energy or whatever else. So anyway, that is the completion of the digestion processes. So the dietary carbs are degraded to monosaccharides, and monosaccharides in the final stage reached the target cells and tissues. What happens after that with the monosaccharides unit, we will see in some other lectures. But before we close today's lecture, we can conclude that the overall process of carbohydrate digestion and absorption is so efficient in healthy individuals, of course, that ordinarily all digestible carbs are absorbed by the time the ingested material reaches the lower duodenum. However, because it's all about the monosaccharides that can be absorbed from our intestinal lumen, any defect in a specific enzyme activity may cause different problems. So, if we have a defect or a deficiency of any of those mentioned enzymes, that causes the passage of undigested carbs into the large intestine. What happens next? What do we have in large intestine? We have bacteria. So bacterial fermentation of the remaining carbs occurs, and those remaining sugars are degraded to 2 or 3 carbon compounds. But what about this is that those products of bacterial metabolism are osmotically active compounds. Plus, you have the formation of a large volumes of carbon dioxide and hydrogen in their gaseous forms. So, the consequence of this is that osmotic disbalance occurs. So our body and our systems are going to react in the way to re-establish this balance. So how is that going to be achieved? You have to remember a little bit our chemistry, inorganic chemistry course. So if you have this balance in osmotic pressure, you're not going to diffuse molecules from one way to another, but you have to bring the solvent. So in this case, water is going to be drawn from the mucosa into the large intestine. And when you're doing this in order to dilute those metabolic bacterial products, so what's going to happen then? Do you have any idea? So if you're bringing water from mucosa into large intestine, exactly, so osmotic diarrhea occurs, and it's exhibited in a number of symptoms like abdominal cramps, diarrhea, flatulence, and so on and so on. So these are some of the problems, and I'm going to give you one example as a conclusion to this lecture, which is a rather common problem, lactose intolerance, in persons with lactase deficiency. So when they're taking lactose-rich food, it cannot be digested due to lactase deficiency. So lactose, dairy sugar, reaches large intestine, causing all the listing problems. So, okay, so this should be all for this first topic. I hope it was okay. I hope it was understandable. And I'm looking forward to seeing you again with a new topic, and we're going to continue with the degradation and catabolism of carbs. So, until the next time and until the next meeting, bye-bye and see you.